हेलो एवरीवन, वेलकम टू बायोमिक्सअप। आज के इस क्लास में हम डिस्कस करेंगे अबाउट द नॉर्दर्न ब्लॉटिंग. सो व्हाई द नॉर्दर्न ब्लॉटिंग इज यूज्ड? इट इज यूज्ड फॉर identifying the RNA molecule, or we can tell that for the gene expression also this techniques can be used. So what is the first process for this northern blotting? So the northern blotting which is present, it is used for identifying the RNA or gene expression. So for that, the first thing which we have to do in this case is we have to extract the RNA from the sample. Then the second step will be that the gel agarose gel, the RNA sample will be run and it will be separated according to the molecular weight. Then it will be transferred to the membrane. After that, the hybridization will take place and that will be detection of the signal. This is the overall thing which we have to understand. So let us see this in a stepwise manner. So this northern blotting which is present, like you can see here, the first step is the isolation of the fragment. Okay, from the cell or the tissue, the RNA is being isolated. Next is the RNA denaturation. So this RNA which is present, it will be first denatured by the help of formaldehyde. Okay, which is with the loading buffer. In the loading buffer only the formaldehyde will be present, because the RNA which is present, it is containing many of the secondary structures with it. So for preventing the secondary structure, it needs to be treated with formaldehyde. Next, in the agarose gel, this denatured RNA will be separated according to the molecular weight. So the large fragment of the RNA will be present in the up region, and the small fragment will be flowed, or it will be separated in the agarose gel and it will be present in the downward region. Okay, next thing, visualization of RNA. Next thing is that we have to visualize that this RNA molecules are present or not. Okay, so accordingly we have visualized this under UV light or in the UV trans illuminator. Next, the next step will be this transfer. This gel is present now. Now we have to transfer it into the blotting paper. Okay, this blotting paper can be liner or nitrocellulose membrane, and this is being done by capillary action. Like in this tray, you will be having the transfer buffer. Upon this transfer buffer there will be filter paper, and upon the filter paper we have to place this agarose gel, and upon that the membrane, that is the nylon membrane or the nitrocellulose membrane, will be present. Up to this membrane there will be paper towel, and we can place a thing which contains the weight. Due to which the transfer buffer which is present in the tray, it will be flowed in the upward direction through the capillary action. Okay, when the towel, when this buffer will be transferred upward, during this time when it will transfer, first it will transfer to the filter paper, then it will reach the agarose gel, then it will reach the membrane. So when this buffer is transferring, by transferring the buffer, it will also transfer the RNA bands which are present in the agarose gel. Okay, after this paper towel, now it is wet, we are sure that the membrane which is present and it contains, now it is blotted. So after that, what we have to do, we have to fix the RNAs in the membrane. So how it is being done? It is being done by UV cross linking. That means we can give the UV rays to it, as well as we can bake it at 80 degrees Celsius. There are two methods of RNA fixation. The first one will be that we can do the UV cross linking. That means we can expose it to UV rays, or else we can also bake it at 80 degrees Celsius. Okay, the next step will be pre-hybridization. So before the hybridization, this process has been done. So what is done in the pre-hybridization? Membranes is incubated in pre-hybridization solution to block non-specific binding sites. For blocking the non-specific binding site, we are keeping the membrane in the pre-hybridization liquid or pre-hybridization solution. Okay, so what are the components of the pre-hybridization solution? It is the then hardened solution, SSC buffer, salmon on sperm DNA, and the blocking reagents. These are the common components. Next, after the pre-hybridization techniques, the hybridization technique is taking place, in which the the RNA molecule which we want to detect, or the genes which are expressing, if we want to detect, if some in suppose in some of the plant there are some of the susceptible genes which are present, and we want to see that in how much expression, how much level it has been expressed, whether it is expressed or not. If we take the wild type plant, there will be low expression or there will be no expression. If we take the susceptible plant, there will be expression of certain genes. So for that type of experiments we can use this. Or else, let us take the example of cancer cells. When there is cancer in a particular cell, some genes will be expressed, and if there is no cancer, the genes will not be expressed. So in this case, this hybridization, northern hybridization technique or northern blotting technique is being used. So in this case, we have to design a probe according to the gene which we are targeting, or according to the RNA which we are targeting. So in that, what happens, the gene or the probe which is present, it is complementary to the RNA sequence. Okay, and this probe which is present, it needs to be radio labeled. Okay, as it radio label, it can be any radio labeling, or it can be fluorescence, it can be fluorescently also labeled. And if there is complementary sequence present in the membrane, then this probe will be hybridized by the help of the hydrogen bond. The next step will be the washing step. What happens in the washing step? There will be multiple amount of probes which we will give, and some of the probes will go and bind with the sequence which are present. So the remaining, they will be present. So the remaining unnecessary things which are present, they need to be washed, and this is being performed in the washing step, in which the removal of the unspecific worm drops are taken place. Okay, the next step will be the detection. Okay, the bound probe is detected using appropriate method depending upon the level. What kind of level we are using, according to that the detection will be done, whether we are using the radioactive probe or non-radioactive probe, or we are using enzyme substrate reaction, or chemical illuminants. According to it, the detection will be done. The next step will be visualization and analysis. Next we will be visualizing the things where the probes will be present. Here we can see the bands, like we can also see whether it is present in more amount or light amount, less amount. Like if we are seeing a light band, that means the expression is low. If you are seeing the bright band, that means the expression is more. In this way, this northern blotting is used for the RNA detection. So these are the type of probes or the type of labels which we can use. The first one will be the radioactive 32P or 32S probes, which are detected by the autoradiography. There are also non-radioactive probes like DIG, deoxygenine and antidic antibody, biotin, streptavidin, fluorescent dye. These are also present, but most commonly we use the radioactive things. Okay, so where are this used? It is used during the gene expression analysis, developmental biology study, disease diagnostics, detection of alternative splicing, mRNA site elimination, study of gene regulation, and detection of specific transcripts. These are the application of this northern blotting. So what are the advantages of it? The advantages is that it is sensitive and specific, because we are using the probes accordingly. So the probes which are present, it will be specific to some of the sequences. So this is the advantage. It also provides information about the RNA size, at what size this RNA is present. When we run in the agarose gel, we can see that what is the size of this RNA which is present. Detect low abundant transcript. If the transcript are also in the low amount, it can be detected, as well as it is helpful in the experimental expression studies. Okay, what are the limitations? The limitations of the RNA is that it will be easily degraded. So here the high quality of RNA is required. Okay, largely it is replaced by RT-PCR and qPCR as well as RNA sequencing. So Water is summary flow. First RNA isolation, denaturation, next is gel electrophoresis, then we will transfer, then fix, pre-hybridization, hybridization, washing, detection, visualization and analysis. This is a process, whole process of the Northern blotting. Thank you everyone for watching this video till the end, and if you like this video, please do not forget to subscribe. Thank you.